I'm Valeria, and I'm going to talk about evolutionary patterns and constraints in measuring pseudogenes kinship. This is how would balance look like if we would put in them protein-coding genes and pseudogenes. Pseudogenes are almost as abundant as protein-coding genes. However, they're annotated as dysfunctional. Pseudogenes arise as copy of the protein coding gene through retrotransposition of mature transcript or through segmental duplication. That's how it's classified in two major classes, processed and unprocessed pseudogenes. They're considered as relics as they often accumulate mutations in the corresponding parental gene's protein coding sequence, and also 75% of them lack any regulatory element. However, what if some pseudogenes are functional? There are thousands of pseudogenes that are differentially expressed in different human tissues. There are hundreds of them that had a detectable peptide as the evidence of their translation. And there are thousands of them that are conserved throughout vertebrates. Currently, the pseudogene's role in genome evolution is considered as the intermediate place in gene's death. But what if pseudogenes can develop a sub or a new function? What if they can evolve in new gene? And what if there is another mechanism of pseudogenes arrival about which we never thought? Currently, the knowledge about pseudogenes' origin is accumulated in the database PsyCube. Knowing where pseudogenes come will help us to know where they go in the future. So PsyCube identifies pseudogenes and the corresponding parental genes by searching in human genome sequences that are similar to the protein coding genes. However, PsyCube contains just the half of currently annotated pseudogenes. Thus, we would propose another approach that will show us the distance between protein coding genes and pseudogenes in a more efficient manner as match distance. Here I would like to talk about difference in PsyCube and match distance approach. PsyCube utilizes the pipeline that is alignment-based, and match distance is alignment-free. The alignment-based approach calculates the exact position of each nucleotide along the sequence, and the match distance utilizes subsequences for much efficient comparison. Alignment-based approach requires multiple parameters which are sensitive in change, and the change may influence drastically the results. However, match distance is more robust as it requires just two parameters, the length of the subsequences and number of them that will be compared. Finally, to find out the distance between sequences, alignment-based approach needs the approximate model of mutation rates, and match distance is based on the statistical model, Poisson distribution, in junction with checkered index. So is match distance good in identifying pseudogene parental gene pairs? As an example, we calculated the distance between CAPDH and its numerous pseudogenes. So here in this graph, we have match distance from 0 to 1. And we see that the distribution of the pairs is centered closer to the smallest match distance. But is it a relevant distribution to say that, well, yeah, those pairs are close to each other? Luckily, CAPDH has only one paralog, and we calculated mesh distance between pseudogenes and paralog 2, we see that this distribution is shifted towards the larger mesh distance, and it means that mesh distance is capable of distinguishing parental level of relationship and more distant relationship. And we may wonder also, is it sufficient enough to identify, to diversify the paralog relationship and unrelated sequences? That's exactly what we did by comparing GABDH pseudogenes to randomly selected protein-coding genes, shuffled sequences, and partially similar sequences. So the only available positive control to verify mesh distance performance is PsyCube. We use it to trace parental transcript pseudogene pairs, and we ran mesh distance on all available pseudogenes and parental genes in PsyCube. There can be two scenarios where mesh distance disagrees with the parental transcript that PsyCube assigned, or it may agree with PsyCube. It appears that it happens almost in half of the cases, so it's hard to evaluate if mesh distance is better or not. To do this, we calculated the mutation density. And what do I mean by that? We take two sequences, in our case, parental transcript and pseudogene, align them, chop them into subsequences, and then compare to each corresponding subsequence, like for how many nucleotides are there different. And finally, we normalize them by sequence length. As a result, we have a graph where on x-axis there's a relative position of compared subsequences, and on y-axis mutation density. So if we would compare two completely different sequences, the graph would have like a straight line on the top. We may see that PsyCube usually matches the pseudogene parental transcript with higher mutation density than mesh distance with lower mutation density. In addition, we see a smiley face that is formed by mutation density of mesh distance, and where PsyCube and mesh distance agree. So at the start and on the end of the sequences, the mutation density is higher, which may indicate untranslated regions more prominent to mutations. And the other part of the sequence has lower mutation density, which we may expect the coding sequence even inside pseudogenes. So mesh distance was capable to identify almost all parental transcripts among pairs present in PsyCube, and the other 1% can be explained with three scenarios. First one, it can be that the pseudogene diverged too much from the parental gene, and mesh distance is not capable to find the relationship. Another scenario is that it's actually the unitary pseudogene, standing for it doesn't have a parental gene in this species genome. And lastly, we may hypothesize that PsyCube could have assigned the wrong parental gene. We noted that pseudogenes without the parental transcript often lie in the gene families with a lot of members, which means that they have a lot of paralogs. And as you may remember from the example with GABDH, mesh distance is capable to differentiate relationship with parental gene and paralog comparing to PsyCube. So along the analysis and finding new parental genes, we may face cases where we can't find the parental gene. our strategy would be first to check what if the parental gene is actually non-coding and it's present in the set of annotated non-coding RNAs. And second step, if first one doesn't work out, would be to assume that the pseudogene is unitary and search for its parental gene in other species. In addition, our pipeline would include the possibility of the pseudogene arriving from another pseudogene. It is theoretically possible if it was segmentally duplicated or retrotransposed in case of transcriptionally active pseudogenes. So let's start from the base. First, we would compare with MASH distance the longest transcript of protein coding genes and pseudogenes. Then, taking the pairs with the lowest MASH distance, we can annotate kind of families with pseudogenes arising from the same parental gene. By comparing with the same MASH distance, them all together, we may find the pairs where pseudogenes are closer to each other than to the parental gene. Let's call such cases pseudogene squared. As an example, we did such analysis on CubeDH and found 3 cases of duplicated pseudogenes. For now, our pipeline is running, and I'll be happy to update you soon with the major results. So CubeDH family is a nice case study, which I was always showing you. It is a homogeneous gene family. It has just 2 protein coding genes, 1 parental, 1 paralog. Most of the pseudogenes are processed and only 1 unprocessed. But what if we would take a look on more complex gene family like CYP? It is a highly adaptive family to changing environment. It has equal number of pseudogenes and protein coding genes and has a variety of pseudogenes types. In addition, the subfamilies of CYP are diverged in the number of paralogs in each of it. It is in particular interesting in case if we question the model of pseudogenization, as CYP family can be a great model to see the pseudogenes contribution to the ongoing evolution. So we performed similar analysis on CYP gene family, and here you see the circle of CYP genes clustered by chromosomes in color, and the links indicate the relationships that MASH distance found. The colored links indicate where pseudogenes are closer to another pseudogenes. We may note that in CYP family, there are even tandem, they're like clusters of pseudogenes arriving from other pseudogenes, or like we would put it here, pseudogene in cube. So in conclusion, I hope I could sparkle your interest to pseudogene with a famous Master Yoda quote. And to conclude the whole talk, we found that MASH distance is effective and robust to identify parental genes and pseudogenes connection, that MASH distance outperforms currently available knowledge from PsyCube, and that we highlighted null evolutionary mechanism where pseudogenes arrive from other pseudogenes. Thank you for your attention.
Very nice talk, Valeriia. And yeah, we are going to have questions.
Hi, thank you for your presentation. I think you have succeeded in making me interested in pseudogenes. I was wondering how similar are they in terms of base pairs? Because I was thinking of the short-read sequencing RNA, which only takes 100 to 300 base pairs. And if they are sufficiently similar, maybe we are not looking at pseudogenes right now, and they're just assigned to the coding genes, but maybe those sequences are already pseudogenes that we can't tell.
Can you please repeat the part about pseudogenes being assigned?
Right. So you said pseudogenes are very similar to their reference parental genes. And right now we do the short read sequencing. It only takes about first 100 to 300 base pairs. If they're sufficiently similar enough, when we do the alignment, maybe the pseudogenes align to the parental genes, and maybe that's what we are counting, and they're already hiding inside the count data.
Yeah, that's a great question. And one of the major problems in studying pseudogenes, not only in like transcriptomics, but also in identifying their peptides. Indeed, they're very similar to the parental genes, and usually in like standard pipelines, they are multi-mapped to the pseudogene and to the parental gene, and such reads are usually discarded. So we underestimate the expression levels of the pseudogenes. And actually, our lab is currently working on this problem. If you'd be interested to talk about more in detail after.
Thank you very much.
Thank you.
Hi, thanks for the interesting talk. I am more interested in the biochemistry of pseudogenes, but I got fascinated by the amount of pseudogenes that we discovered in different families. My question was, did you find any kind of correlation between the number of substitutions in the pseudogene with their expression levels?
Interesting question. Well, linking to the previous question, it would be sometimes difficult to make a good analysis, right? Because we don't always know if this is expression of pseudogene or not. So I think this is a good question for further research. But also the number of mutations don't necessarily indicate that the expression will be lower. Maybe the sequence then transformed to something very new, like a new gene. So thank you. Definitely
It's
look at
just
that.
very interesting because I never realized that there were so many pseudogenes in the human genome.
Yeah.
Thanks.
Thank you.
I think it's a very hard problem. So good for you. Did you investigate or use as part of the information that can help inform what you're seeing, how they're organized on chromosomes? Are they, do you see them on the same chromosome, different chromosomes? Is there a tandem organization? I mean, you are using processed pseudogenes, so you're using that information, but are you using other spatial information?
We're using processed and unprocessed pseudogenes. Taking into account all of them, we don't see the correlation, like the conglomerates of pseudogenes around the genome, like clusters. Sometimes
don't see clusters.
I mean, there is no strong correlation. If we go by gene family level, we might find some specific examples. In particular, the C family that I presented today, some of the subfamilies are clustered. So we may find even that there's pseudogene. That would be interesting to go deep to. Thank you.
Yeah, I mean, I would think you would see the processed pseudogenes would be much less likely to be in tandem arrays than for example.
Possibly. Thank you.
Tell us next year.
Hi, thanks. That was very exciting talk. I was just wondering about, have you seen much relationship between sort of the conservation of specific subsequence of the parental gene and the pseudogene? Like, are they corresponding generally, or do they kind of start evolving in different ways?
That's interesting question. Well, in general, I didn't see the precise analysis of the question that you posed. And it depends also like on the age of the pseudogene, because sometimes they're very new and we cannot see like the correlation where it goes. But we may also anticipate the coevolution between some pseudogenes and parental genes, which will be interesting to look at. Thank you for your interesting question.
Yeah, we'll come back to you next year on that too. Thank you.